Welcome, students, back to Chapter 9. So in a previous lecture, we discussed how muscle tissues are one of the tissues that are excitable, that are able to change their membrane potential, that like nerves, they can depolarize by having this influx of positively charged ions. Understanding that is important because how our muscles contract that allow us to move and to breathe and to talk is dependent on that ability of the muscle tissue to be excitable. If they were not excitable tissues, they would not have this inherent ability to take chemical energy and transduce it into mechanical energy via movement. So the contraction of a skeletal muscle fiber is honestly amazing to me. It is this series of steps that have to occur kind of like dominoes falling down, one after another in sequence in order for us to walk, to talk, to breathe, to gesture, stuff that we don't even really think about, we just do. And our muscles can do that through this kind of contraction process. So it starts with nerve stimulation. Skeletal muscle is voluntary, so it does have to get a kind of little stimulus from the nervous system. We then have to generate an action potential along the sarcolemma, the muscle membrane. That action potential must kind of propagate along the muscle membrane down deep into the muscle fiber itself through the T-tubules. That leads to the levels of calcium increasing inside the skeletal muscle cell. That calcium acts as kind of an inhibitor, or it takes away the inhibitor. So we get what we call a crossbridge, a connection between our thin and thick filaments, and then the filaments slide past each other and overall shorten each sarcomere, which then leads to an overall shortening of the skeletal muscle fiber itself. So steps 1 and 2, nerve stimulation, action potential generation in the muscle, that takes place at the neuromuscular junction, where the nervous system connects to the muscle. Steps 3 and 4, the propagation of the action potential, the increase in calcium levels, is what we refer to as excitation contraction coupling, because it links the electrical events, excitation, to the mechanical events, the actual contraction. Calcium is a key figure in excitation contraction coupling. And then all of the rest of it, once we're past that kind of nerve part, we're now in the muscle. So excitation contraction coupling takes place in the muscle, as does crossbridge formation and those sliding filaments. And so really it's kind of a couple of maybe 5 or 6 kind of broad steps, but we're going to break it down into these kind of 3 pieces: neuromuscular junction, excitation contraction coupling, and crossbridge formation. And we're going to actually look at the 20 or so steps that have to happen in sequence in order for us to, just wave to our friends. So it's kind of 2 phases roughly. Again, action potential arriving at the nervous system, changing the membrane and depolarizing the membrane, igniting an action potential, the action potential traveling along the muscle membrane, traveling along the T-tubules, leading to the release of calcium, which then snags on to the troponin, pulls the binding sites kind of free. We get our crossbridge formation, and we get those sliding of the filaments. There are 3 figures that we're going to walk through associated with this process. There are also then 3 videos where it shows you step by step what is occurring. And I find the videos really, really, really helpful because I love figures. I love looking at diagrams. I think diagrams are really helpful, but if I can see stuff moving, that's great for me. I love to see stuff moving. So definitely watch the videos. We're going to start at the neuromuscular junction. So the type of neurons that connect to skeletal muscles, that stimulate skeletal muscles, are what we call somatic motor neurons, which is just really kind of a fancy word for saying they're voluntary motor neurons. If you want to do a high kick, you have to choose to do that high kick. It's a voluntary action. The axons of these nerves, of these neurons, extend from the spinal cord out to the skeletal muscle. And as they get into the skeletal muscle, they divide into all of these little branches that go out to each little fiber within the muscle. And so that neuromuscular junction is the connection between the neuron and the muscle. Each skeletal muscle fiber gets one neuromuscular junction, typically situated about midway along the length of the muscle fiber, so that the action potential can kind of travel out in both directions. The anatomy of the neuromuscular junction consists of kind of three pieces, really. We've got the axon terminal, the end of the axon, what we call this synaptic cleft. It's this little fluid-filled space, and then the motor end plate of the muscle. So the action potential starts here, gets transferred through the synaptic cleft using some neurotransmitters, and then kind of stimulates the muscle fiber. The nerve and the muscle fiber don't touch the end of the axon terminal. There is this kind of space between them. So if we look inside the end of our axon terminal, we'll see lots of little vesicles containing a neurotransmitter. If we look at the neuromuscular junction, you'll see it'll have these kind of folds in it. It's not like a smooth surface typically, because by having these kind of folds, we're allowed to have, we can have more surface area, which means we can have a lot more of these receptors that are going to receive the neurotransmitter. And the neurotransmitter that our skeletal muscle uses and our somatic motor neurons use to activate our skeletal muscles happens to be a neurotransmitter called acetylcholine. So here is our motor neuron. Here is, you can see how it kind of divides into these branches. Each muscle fiber will get one neuromuscular attachment. And we're going to zoom in on one of those little neuromuscular junctions. So you can see here is the axon terminal. Here is the fluid-filled synaptic cleft. And then here are the junctional folds in the sarcolemma of the motor end plate. By having the folds, you're able to have more receptors for our neurotransmitter acetylcholine. So when you decide to activate a particular skeletal muscle, your brain sends a signal out to the spinal cord, which then sends a signal out along this motor neuron, and the signal, the action potential, arrives here at the axon terminal. Well, when the membrane potential of the axon terminal changes, that opens these little voltage-gated calcium channels in the axon terminal. Calcium then influxes into the axon terminal. That influx from outside the axon, that influx of calcium causes the release through exocytosis of the acetylcholine, which basically then just kind of diffuses across the synaptic cleft and binds onto these little receptor channels. When acetylcholine binds onto the receptor channels, the channels open up. We get this influx of sodium, a little outward flux of potassium, but we have much greater influx of positively charged sodium than we do outward flow of positively charged potassium. They're both positively charged. But so even though we're going to lose a little positive charge, we're gaining just gobs and gobs of positive charge in the form of sodium, and that creates what we call a end plate potential. It depolarizes this particular part of the muscle membrane. So this particular part of the muscle membrane gets depolarized. The signal then spreads this way and this way. And that leads us into the second kind of set of steps that have to do with excitation, contraction, coupling. So once we have our end plate potential, we have to again get it to spread. So that spreading is this kind of depolarization that travels along the length of the membrane. So we created our end plate potential through the stimulation from the nervous system. That depolarization wave then spreads outward, both directions. We're only looking at one direction here. As that depolarization spreads along the muscle membrane, that will open ion channels for sodium. They happen to be gated ion channels for sodium, voltage-gated ion channels. Sodium influxes into the muscle that continues to spread the action potential. As the wave of the action potential kind of passes, potassium channels will open up, potassium will leave, and this particular part of the membrane will then kind of repolarize back to normal. Let's see what that looks like. So you can see we're at a part of the membrane where it's just resting. The propagation kind of spreads. The sodium channels open. That depolarizes us. Sodium channels close. Potassium channels open. And that repolarizes this particular part of the membrane. And then eventually the potassium channels will close and will kind of just reset. But the propagation and the action potential and the depolarization continues to spread and spread outward from the muscle membrane and from that neuromuscular junction. So this excitation, contraction, coupling links all of the kind of electrical currents, the excitation, to the actual mechanical movement and sliding of the filaments. That's the contraction part. So excitation, contraction, coupling. That action potential spreads along the muscle membrane, spreads along the sarcolemma, and then down deep into the muscle fiber through the T-tubules. Remember, the T-tubules are essentially just indentations of the muscle membrane going deep into the interior of the muscle. Well, those T-tubules have voltage-gated calcium channels. And when the action potential hits those voltage-gated calcium channels, they open up, and sarcoplasmic reticulum access is granted, and calcium rushes out of the sarcoplasmic reticulum and into the intracellular fluid of the muscle cell. When you have low concentrations of calcium intracellularly, the tropomyosin is wrapped around the actin, and you can't get the myosin heads to attach to the actin. At that point, the muscle fiber will be relaxed because we can't make any cross bridges. But when that calcium comes out of the sarcoplasmic reticulum, the calcium will bind onto that kind of anchoring troponin protein. The troponin will change shape. That will unwind the tropomyosin away from the binding sites on actin for the myosin. Myosin can then make a cross bridge with actin, can kind of grab on, and we can start the sliding of the filaments, which leads to the shortening of the sarcomere, which leads to the contraction of the muscle fiber. When we're done with our particular movement, when nervous stimulation stops, then the calcium releases from the troponin and is essentially actively pumped back into the sarcoplasmic reticulum, which requires ATP, and contraction of the muscle fiber will end. So let's take a look what that looks like. So let's go back a step. We had an action potential in our motor neuron that led to the release of a neurotransmitter, which led to the depolarization and the end plate potential at the motor end plate of the muscle fiber. That action potential spreads, and it spreads along the sarcolemma and down the T-tubules. As the action potential spreads down the T-tubules, these calcium voltage-gated ion channels will open up. That releases calcium out of the little terminal cisternae of the SR and into the intracellular fluid of the muscle fiber. That will allow calcium to bind onto the troponin, which is currently kind of wrapped around and locking the tryptomyosin into place, blocking the sites. Calcium binds onto troponin. Troponin changes shape. The tryptomyosin unwinds from the actin, and we now have free sites for myosin to make a crossbridge, a connection with the actin fibers. And we now have a crossbridge. We can now start the sliding of our filaments. Crossbridge formation and kind of reformation is cyclical. This particular process will occur as long as calcium levels are high and the binding sites on the actin are free for the myosin. So it really starts actually with what we call cocking. It's essentially energizing the head of the myosin. We need to hydrolyze and split an ATP molecule to put myosin in kind of the perfect kind of active, energized formation. Once the myosin has been activated, then a crossbridge can occur. The head of the myosin will kind of grab onto the little actin protein, and we get our nice little crossbridge. The next step is what we call the power stroke. The myosin head essentially pivots and bends. It's called the power stroke, the working stroke. It's like rowing, right? And that slides the actin filament kind of 1 unit towards the middle of the sarcomere. An ATP molecule, another one, will then attach to the myosin head. That will break the cross bridge. The myosin head will be re-energized by splitting the ATP, and the myosin will then grab onto the next actin molecule in the chain. Power stroke, crossbridge breaking, activation, crossbridge formation, power stroke, detachment, over and over and over again. And the myosin grabs onto one actin and then the next actin and the next actin and the next actin, and they kind of ratchet the filaments towards the middle of the sarcomere. That ultimately shortens the sarcomere, which then, as we get more and more overlap in the filaments, which then, as each sarcomere shortens, ultimately leads to the contraction of the skeletal muscle fiber. And as each skeletal muscle fiber contracts, ultimately the muscle will contract as well. So that's muscle contraction. 20 so steps that have to happen in this proper sequence. We've looked at the diagrams, we've walked our way through it. Make sure that you go watch the videos that are in the study tools folder that will look at these 3 parts: neuromuscular junction, excitation, contraction, coupling, and crossbridge formation. If you are misunderstanding this at all, those videos are by far your best resource.